Our brains are the single most important organ in our bodies, and they run very similar to how a car does. Cars have tubes and valves that transfer gas from the gas tank to the engine, while blood vessels transport oxygen and other nutrients to our brains via red blood cells. However, if you put regular gas into a car that requires premium, engine failure may occur. This is similar to how sickle cell disease affects the brain. Sickle cell disease is characterized by red blood cells with a sickle shape. Sickle red blood cells are inefficient at carrying oxygen and tend to clump together, causing them to flow differently than normal red blood cells do. This change can damage blood vessels, forcing the vessels to change shape, also known as vascular remodeling. These vascular changes can lead to further life-threatening complications like stroke or aneurysm because the brain cannot regulate vascular remodeling like it could in someone without sickle cell disease. Despite the severity of these complications, there's currently no way for us to view blood flow in the brains of adults with sickle cell disease and provide preventative stroke treatment like there is for children. Since it is well documented in scientific literature that geometry affects the flow of blood and vice versa, my project explores whether this relationship between form and function can be applied to patients with sickle cell disease to indicate a risk of stroke. I specifically focus on the circle of Willis, which is a major artery system that sits at the base of the brain and branches out to provide blood to the rest of the brain. Circle of Willis is where researchers tend to see changes in blood flow and shape in cerebral blood vessels. I first create patient-specific models of the circle of Willis based on MRIs of healthy control patients, patients with sickle cell disease, and sickle cell disease patients who have had a stroke. Patient specificity is important because everyone's cerebral vascular geometry is different, so certain geometric variations may not be the result of sickle cell disease, but can still impact blood flow and further encourage or inhibit stroke development. Comparing sickle cell disease patients' circle of Willis anatomy with control patient anatomy will help elucidate whether certain geometric patterns are part of the pathology of the disease or otherwise. By analyzing geometric parameters like tortuosity and curvature that have been associated with altered blood flow, we can theoretically identify and quantify these geometric differences that could lead to stroke. Our future direction is to use these models to run computational simulations of blood flow with boundary conditions specific to the patients. I will then compare results from the simulations to the geometric parameters I find to see if there's a correlation between specific changes in blood flow and the circle of Willis geometry of adults with sickle cell disease. This process will be a non-invasive method of analyzing the geometry of cerebral vasculature that will hopefully elucidate the pathophysiology of stroke in adults with sickle cell disease and be a basis for developing preventative treatments rather than only offering palliative care. Thank you.